All right, ladies and gentlemen, welcome to the first of our quarantine videos. No, I'm kidding. So what we're going to learn about is the remainder of protein synthesis, the second major step in the process called translation. And here's just a little lovely nerd humor for you to absorb. So let's talk first about, as we usually do, the differences in this process between eukaryotes and prokaryotes. So prokaryotes, remember, the major big difference they have is there's no nuclear membrane surrounding their DNA, which means that while an RNA is being produced by this lovely little RNA polymerase here, it can, at the exact same time as it's being produced, be translated by ribosomes. So you'll notice that these ribosomes are all translating an mRNA that has not been released from the RNA polymerase yet, that's not completely done being transcribed. Though keep in mind that this means that you can't have any processing of mRNA happen before you can start translating because it's basically getting translated as it's transcribed. This speeds up the process, so bacteria can conceivably go from, okay, a signal specifying let's create a gene or let's create a protein, all the way to protein produced in a shorter time than eukaryotes. But like I said, they have less control over the process. In eukaryotes, translation is a process that happens exclusively in the cytoplasm, which means that first, an mRNA, after it's produced, has to get out of the nucleus before it can be translated into a protein. And that's why, if you'll remember from our transcription lesson, that methyl G cap and that polyadenylation sequence are important because they help get that mRNA that's mature and ready for transcription to exit the nucleus. Once it's exited from the nucleus, then it can so what'll happen is the next step is a ribosome will start to assemble around that mRNA. And then based on some signals at the beginning of the mRNA, that ribosome will either stay free floating in the cytosol or it will bind to the nearest rough ER. And the reason why that happens differently, that translation happens differently in those two cases, is something we'll talk about a little later on. So just a general overview, you start out with an mRNA transcript, and it is synthesized in one long chunk. So if you were looking at this in an actual molecule, it's not physically divided up into these codons. What happens is a molecule comes along and reads it 3 base pairs at a time. So it will read these 3 letters, then it will read these 3 letters, and then it will read these 3 letters, and so on and so forth. And we'll talk about how you can tell which amino acid is coded for by which codon, but in general, as long as you have the translation chart, the AUG in an mRNA always codes for a methionine. So wherever you have AUG, that is going to be where a methionine is going to be found in the protein that's produced. This GGC codes for a glycine. So where you see GGC, you will have a glycine in the mature protein. The UCC codes for serine, AUC codes for isoleucine, GGC codes for glycine, GCA codes for alanine, and you'll notice there's nothing after that because we have a stop codon at the end. It doesn't signal another amino acid. What it signals is this protein is done, and it allows the entire ribosome to kind of fall apart and everything. So another important thing is note that we are forming peptide bonds during this process. Remember, peptide means protein. So let's talk about the different steps in translation. Initiation is the first step, and what happens is the ribosome assembles around an mRNA start codon, which is AUG. Now, notice that this is a codon that also stands for the amino acid methionine. What this means is that every single protein that's produced in the body, right after it's produced by a ribosome, starts out with a methionine. And every single mRNA transcript, the part that's actually being translated, is always going to start with an AUG. Notice a few things. There are binding sites where the mRNA binds to the various units of the ribosome, notice it has a small subunit and a large subunit. So the small subunit tends to bind to the mRNA, and then a tRNA gets associated with the start codon. This tRNA is called an initiation factor, and it's going to start the process of translation, and it's got a methionine attached on the other end. Over here, what happens next is the large subunit of the ribosome will attach on top of that, and then this process is ready to get started. By the way, the rRNA in the ribosome, you'll notice, interacts with the mRNA, and that's going to help with recognition of an mRNA attaching to a ribosome, so that way it can get translated. You'll also notice that there's a sequence of mRNA before you hit that start codon, and that sequence of mRNA is there to basically help it associate with the ribosome. So it's kind of like a, hey, look at me, I'm a sticky end attached to me ribosome. The next step is elongation. Elongation is going to take as long as it takes to produce a protein of a certain length. So how do we know which codon or which mRNA codon stands for which amino acid? Well, luckily for you, scientists have done all of this work to figure out which amino acid codes for which codon. And they put it all together in this nice handy dandy chart that probably will haunt your nightmares after any genetics class. Each of the 64 possible codons that you can make, and a codon is 3 letters, so there are 64 possible codons that can be made. Every single codon that can exist is on this chart. And so each of these represents either an amino acid or a stop codon. So notice we have 4 highlighted amino acids, AUG, which is the start codon, also is the amino acid methionine, and then these 3 up here, which are each stop codons. So whenever you see that in an amino acid sequence, what you are looking at is a signal to stop translation. So let's go ahead and see what's going on. If we have a codon like UGC, how do we read this chart? Well, notice up at the top that there's a section that says first base. You're going to look there for the first base in your codon, which is a U. Okay? That means you are going to focus only on stuff within this box, and if you'll look, you'll notice every single codon in that box starts with a U. The second base, which is at the top, in this case is a G, which means we're only going to look at stuff in that column. If you'll look at that column, every single codon in that column has G as the second base. So we're going to focus on only that one box where U is the first base and G is the second base. Now, most of the times, if you do have a codon actually listed out here, you can just look at that and figure out, oh, hey, UGC is cysteine. But sometimes they won't put the codon on the chart, and this is just for other classes on the AP test. They'll always have a specific chart, which I'll show you on the next slide. But the third base is over here. Our third base was C, which means we're going to look in this line. So where all 3 of those boxes intersect is where we are going to find our codon and our amino acid. So that tells you UGC will always code for an amino acid of cysteine. So if the mRNA codon has UGC, that means that at that location, you'll have a cysteine amino acid. So this is the actual chart that will show up on the AP test whenever you have a question that needs it. Notice it's fairly similar to the other chart. The main difference is that you'll notice things are kind of grouped, and you'll also notice that you don't have the full name of the amino acid, but that's okay. You don't have to memorize an amino acid code. All you need to be able to do is read this chart and figure out, okay, if I have this codon, what amino acid is it going to code for? So let's talk a little bit about the fact that you can group these. So what I've done here is I've kind of, not quite prettily, but I've basically color-coded this codon chart. There are 64 possible codons, and there are 20 amino acids that need to be coded for. If you add to that, there's also the stop codon that you need. But if you'll look, there are some amino acids that have more than one codon that codes for them. So for example, leucine, which you'll notice right here, has 6 different codons that code for it. However, tryptophan only has 1 codon that codes for it. This is called redundancy, the fact that you have multiple codons coding for the same amino acid. There is a reason for this, and the reason is that it's going to allow you to have the same amino acid coded for it, even if you have a mutation. So let's look at an example. AUU. AUU is this guy right here. So it codes for an isoleucine. Well, you can have a mutation that changes the third base in that codon to AUC or AUA, and it still will not change the amino acid that's produced in that spot, which is a very good thing because, most mutations are either harmful or, some of them don't really have an effect. And so you don't want to have a harmful mutation develop. Let's look at UUA. So UUA is a leucine. And if it gets mutated to a CUA, that's still a leucine. Now, notice there are 2 amino acids on here that only have 1 codon that codes for them, methionine and tryptophan. So for those, you need to make sure that you don't have any mutation in those codons. Otherwise, you risk ruining a start codon or just completely screwing up the amino acid that's there. All right, so that's why redundancy is a very important feature of the genetic code. Another important feature of the genetic code is it is universal. Most organisms use that whole codon chart that we just looked at exactly as it's seen. There are a couple of bacteria that have 1 or 2 variations where one of those stop codons will code for an amino acid instead of coding for stop. But other than those couple of species, nearly all organisms on the planet use that same translation chart. Same translation code. Another thing, all organisms use the central dogma of genetics. That's how they produce protein. So information flows from DNA to RNA to protein. All organisms have their genetic code read 3 nucleotides at a time. And each of those codons, made of 3 nucleotides, represents a single amino acid. And then what does this mean? Well, this is a sign that all organisms share a common ancestor. If we don't have variety in such a basic fundamental process, then it was probably inherited from a common ancestor. And a really cool consequence of this is that you can actually take a gene from one species, transfer it to another species, and it will produce the same protein. So this is how, for example, we're able to take bacteria, introduce the gene for human insulin into them, and have them produce insulin. It's also responsible for this very neat example, what you see up here. These are mice pups that have been induced to produce a protein that is originally found in jellyfish. It's called green fluorescent protein, and when you shine UV light on them, they glow green. No, they don't glow in the dark, but it's still pretty neat that you can make glowy mice. Now, what is the application of this if, you're not going to see glow in the dark mice? Well, the thing is we can use this GFP gene as a marker and use it to try and figure out things that are happening inside of an organism. Imagine, for example, if you could make all of the cells in an organism that produce a certain protein glow, then you can track where different proteins are translated. So it's a really cool consequence. The actual thing that does the translating directly is a tRNA. What you'll notice is that on one end, it's got what's called an anticodon that pairs with an mRNA codon, and then it's got an amino acid binding attachment on the other end. Depending on the anticodon it has on one end, it will have a specific amino acid on the other end. So inside of a cell, you don't have that translation chart. What you have are tRNAs. And so the tRNAs are going to be what directs the amino acid pairing with a specific RNA codon. Let's look at this process. So let's say you have a tRNA that's already like one of them's being ejected, and then we've got an RNA moving over. So ribosome shifts over, so that way you can get to the next codon. A new tRNA is going to enter that site in the ribosome. You don't need to know the names of the sites or anything like that. And it's going to base pair to the mRNA codon. Based on the mRNA codon it has, it's going to have a specific amino acid attached to it. And then the ribosome is going to take whatever amino acid chain it's already been producing, and it's going to bind it to the amino acid on that new tRNA. So you have this growing polypeptide chain, this growing chain of amino acids, and it is going to basically be transferred to the next tRNA. Notice that that step does take energy because you are forming a chemical bond. After that, you have a tRNA that has a growing polypeptide chain attached. And then what's going to happen is the ribosome is going to shift over until you get to the next codon. That old tRNA that no longer has the polypeptide chain attached to it gets kicked out. And this whole process repeats over and over and over. Also notice there are multiple steps that use energy. So if you don't have energy in a cell, you cannot synthesize proteins. And then finally, you get to termination. At the end of translation, what's going to happen is you get to a stop codon. Well, the only thing that matches a stop codon is this release factor. The release factor, when it binds, has no amino acid attached to it. There is nothing for the ribosome to attach that polypeptide chain to. And so when it tries to, or when that release factor binds, it triggers the dissociation or disassembling of the ribosome. So that whole complex just kind of falls apart. The ribosomes pieces are now able to go translate another mRNA. That mRNA can be translated by another ribosome, and then that release factor can be used to stop translation of another protein. And so this whole cycle can start going over again. It's great because you can reuse all of these parts. You can use the mRNA to make a lot of copies of one protein based on how many times it's translated by several different ribosomes. And, you don't have to use the energy to make new ribosomes, make new mRNAs, make new release factors, make new tRNAs in order to have this process continue. After translation, you can have a number of different modifications happen to a protein. In some cases, they'll cut off, so they'll like cleavage. They might cut off that beginning methionine because it's not needed. They might modify the amino acids on the polypeptide chain. They might add some chemical groups, and all of this is designed to make that protein fully functional. And then finally, you need to get that protein to its final target. This is why it's important where the ribosome that translates the protein is found. If that ribosome is free floating in the cytoplasm, in the cytosol, then that means that you're probably going to target the protein that's made to another organelle, or it's going to be functioning in the cytoplasm. On the other hand, if a bound ribosome bound to the endoplasmic reticulum is producing that protein, then it's either going to end up in part of the endomembrane system, so stuck in the membrane, or it's going to end up either being secreted, like so, or it's going to become part of the plasma membrane. And which of those happens kind of depends on some signaling sequences in the protein that will direct it so that way the cell can transport it where it needs to go. All right, that is the end. Our next lesson is going to be regulation of gene expression and